For centuries, leprosy was called a disease of the tropics, a disease of Asia. That was wrong. The answer had been hiding in the ground for 1,000 years, waiting for someone to look. Europe's medieval leper colonies held thousands of people, and for most of history, nobody thought to ask where the disease actually came from. The bones were speaking, but nobody had listened for a very long time. Across Europe, leper cemeteries held thousands of bodies, each one a record waiting to be read. Then scientists tested the bones, and the answer reversed everything. Some of these skeletons had been buried alone, separated from their communities, even in death. Leprosy is caused by a bacterium called Mycobacterium leprae. It attacks the skin, the nerves, and the bones. In advanced cases, it leaves unmistakable marks on the skeleton. The nasal bones collapse. The fingers and toes shorten. Archaeologists can identify leprosy victims from their skeletons alone, centuries after death. But identifying the disease was not the same as understanding where it came from. The fingers told the same story, shortened and reabsorbed by the disease. Healthy bone versus leprous bone. The difference was unmistakable after 1,000 years. For most of medical history, the assumption was simple. Leprosy came from Asia. European colonial doctors in the 18th and 19th centuries found leprosy endemic across India, Southeast Asia, and the Pacific Islands. The Crusaders returned. The lepers multiplied, and Europe blamed the East. That was the story for centuries. They assumed the disease had always been there. European crusaders or traders, they believed, had brought it back in the Middle Ages. The narrative was tidy. Asia was the origin. Europe was the victim. The Crusades were the bridge. The Crusader ports were melting pots. Diseases traveled as easily as spices. In medieval Europe, lepers were cast out, forced to carry bells to warn of their approach. They lived in lazar houses on the edges of towns, treated as if already dead. But in 2013, a team at the University of Tübingen changed everything. They tested the assumption with ancient DNA. They extracted bacterial DNA from medieval bones, and the results contradicted 2 centuries of medical orthodoxy. Schoenemann classified the medieval strains into multiple distinct lineages. Europe had them all. The team was led by Verena Schoenemann. They collected skeletal remains from medieval leper cemeteries across Europe. Denmark, the United Kingdom, Sweden, the Czech Republic, samples spanning 5 centuries of European leprosy. From these bones, they extracted fragments of Mycobacterium leprae DNA, fragments that had survived for up to 1,000 years. The bacterial DNA was degraded but recoverable. Ancient DNA techniques had improved enough to reconstruct near-complete genomes from medieval pathogens. When Schoenemann's team compared the medieval European strains to modern leprosy strains from around the world, the result was a shock. Medieval Europe had the most genetically diverse leprosy in the world. Extracting bacterial DNA from 1,000-year-old bones required dozens of amplification cycles. Each bone sample produced millions of DNA fragments. Most were human. A tiny fraction were bacterial. Separating the pathogen signal from the human noise was the hardest part of the analysis. The direction of spread had been assumed for 200 years. The genomes said it was backwards. If leprosy started in Asia, Europe should have had less diversity, not more. It had more. In population genetics, diversity means time. The longer a pathogen circulates in a region, the more genetic variants it accumulates. If Asia were the origin, Asian strains should be the most diverse. They were not. Europe and the Near East carried strains from nearly every major lineage of Mycobacterium leprae. Asia carried a subset. The same pattern you see when a pathogen migrates outward from its homeland, losing diversity as it spreads. The direction was backwards. Leprosy didn't come to Europe from Asia. It may have gone the other way. 2 centuries of medical orthodoxy overturned by medieval bones and bacterial genomes 500 years, the same strain, still alive in a British forest. That is how slow this bacterium evolves. The team knew the result was controversial, but the data left no room for the old explanation. But the story got stranger, because the medieval bones were not the only surprise. In 2016, researchers in Britain tested something nobody expected to carry leprosy. Red squirrels, the small native squirrels found across the British Isles. A team led by Anna Mendham at the University of Edinburgh made an unexpected discovery. They tested tissue samples from red squirrels on Brownsea Island in Dorset. The squirrels showed skin lesions. The researchers tested them and found Mycobacterium leprae, not a close relative, not a similar species, the same pathogen that causes leprosy in humans. British squirrels were carrying a medieval strain of leprosy, still alive, still circulating. The squirrel strain was genetically almost identical to strains found in medieval British leper bones. The bacterium had survived in an animal reservoir for 500 years after leprosy vanished from British humans. The squirrels were a living archive of medieval disease. A bacterium that vanished from human populations was still hiding in the forests of southern England. Brownsea Island, a nature reserve in Dorset, home to one of the last red squirrel populations in Britain. The squirrels showed no obvious symptoms. The disease was hiding inside apparently healthy animals. The Near East was a crossroads. If leprosy originated here, every trade route could have carried it outward. Only under the microscope did the medieval passenger reveal itself. In 2018, Schuenman published an expanded study. More bones, more sites, more centuries covered. This time, the dataset included ancient strains from the Near East, Central Asia, and East Africa, alongside the European samples. The phylogenetic tree grew more detailed, and the picture became clearer. The deepest root of the tree sat between Europe and the Near East. The criticism was published in a top journal. Schuenman responded with additional data. The debate continued. The oldest divergence events in the bacterium's evolution occurred in that region. The oldest strains were European and Near Eastern. The Asian strains branched off later. Molecular clock estimates suggested thousands of years of diversification in Western Eurasia, long before the bacterium appeared in South or East Asia. The trade routes that had supposedly brought leprosy to Europe may have actually carried it away from Europe. The Silk Road carried goods both ways. It could carry disease just as easily. Traders, soldiers, and pilgrims moved along these routes for millennia. Pathogens traveled with them. Indian researchers are now building their own ancient DNA dataset. The answer may become even clearer. But this is where the story gets more nuanced, because not everyone agrees. Ancient DNA rewrote the direction, but it didn't close the case. Some researchers argued the European diversity could reflect sampling bias. More European bones had been tested. If Indian or Chinese leper cemeteries were excavated with the same intensity, would their strains prove equally diverse? That is a fair objection, and it hasn't been fully answered yet. Indian researchers, led by Pushpendra Singh, tested ancient skeletal remains from archaeological sites in Rajasthan. The Indian strains were genetically younger, fewer mutations, less internal diversity than the European medieval strains. This was consistent with the European origin model. The Indian strains looked like they had arrived more recently, but Singh's team also noted that leprosy has been described in ancient Indian texts dating back over 3,000 years. The shared immune response between TB and leprosy was not a coincidence. The two bacteria are evolutionary cousins. Historical texts described leprosy in India long before the Crusades. The disease was present in Asia far earlier than the colonial assumption of a Crusade era introduction. The Indian evidence was real but younger. Ancient texts described leprosy, but DNA told a different timeline. Archaeological evidence confirmed leprosy in India by the first millennium BCE, but European strains were still older. So leprosy was old in both places. The question is, which was older? And genetics says Europe. Molecular clock analyses dated the ancestor of all known strains to roughly 4,000 years ago. There is also the question of how leprosy disappeared from Europe in the first place. By the 16th century, leprosy had virtually vanished from Western Europe. Nobody fully understands why. The WHO declared leprosy eliminated as a public health problem in 2005. The bacteria disagreed. Some theories point to improved sanitation, others to competition with tuberculosis, which may have provided partial cross-immunity. The tuberculosis hypothesis is compelling. As TB spread through crowded cities, populations developed immune responses that also attacked leprosy. Living conditions in medieval cities were perfect for tuberculosis, and TB may have accidentally driven leprosy out. In some medieval cemeteries, leprosy and tuberculosis victims lay side by side. The diseases competed for hosts. Research continues in the countries that still carry the burden. The science is catching up to the suffering. Modern experiments show that TB vaccination provides partial protection against leprosy. The cross-immunity is real, but the ancient DNA added another possibility. The European strains may have evolved to become less virulent over time. Pathogens that kill their hosts too quickly have a problem. They run out of hosts. Mycobacterium leprae has the slowest doubling time of any known human pathogen, 14 days to divide once. The bones go back into storage, but the data they yielded has already changed the story. This extreme slowness means the bacterium evolves at a glacial pace. Its genome has barely changed in 1,000 years. And that near-frozen genome is what makes the red squirrel discovery so remarkable. The squirrel strain and the medieval human strain are almost identical. After 500 years of separation, the combined evidence from bones, bacteria, and squirrels tells a single story. The disease came from here. The blame went there. And it took a 1,000-year-old bacterium to set the record straight. A story that reverses the old narrative entirely. Leprosy was not a disease that Europe caught from Asia. It was a disease Europe gave to the world. Trade routes, colonial expansion, and migration carried European strains eastward and southward over millennia, and the name itself carries the stigma. For centuries, leprosy was associated with poverty, the tropics, and the global south. That framing was not just wrong. It shaped policy, funding, and treatment for generations. Today, leprosy still affects over 200,000 new patients each year, mostly in former colonies. The disease that Europe blamed on Asia may have been Europe's own export. The bones told the truth. The question is whether the history books will catch up. Leprosy is treatable today. A 6-month course of antibiotics cures it. But stigma remains. The cure has existed since the 1980s. The misconceptions have lasted much longer. 200,000 people are diagnosed every year, most in countries that were once European colonies. For 1,000 years, leprosy victims were isolated, stigmatized, and stripped of their rights. Their bones sat in the ground for centuries. And when scientists finally tested them, the bones revealed that the story everyone believed about where this disease came from was wrong. If this changed what you thought you knew, subscribe. There is always more buried in the evidence. Some truths survive in the strangest places. You just have to know where to look.